professor at the Department of Genetics, University of Delhi, South Campus. I am primarily teaching human genetics to the students at the postgraduate level. My research focus is primarily to understand the different genomic alterations that underlies the cancer genome. And we try and make sense out of the nuances that underlies the cancer genome, and then we pair our discoveries with functional analysis. Develop mouse models, we try and validate the different genetic alterations that underlies the cancer genome, and we hope for to identify major or therapeutically relevant vulnerabilities that can be translated to the clinics and essentially towards the betterment of the patient care in India and across the globe. One of the interesting study from my former postdoctoral group by Matthew Meyerson, what they talked about is that your ancestry, the global ancestry, is way too far more important and significant than your environmental ancestry, or I think they talk about if an Indian migrates from India, well, the study was done on Latin Americans, but I would imagine that that would be extendable to the Indians as well. And if to migrate from India to the US or to any of these countries, their incidence or their likelihoodsness of getting lung cancer will be more defined by their genetic ancestry that as an Indian, not just because they have kind of moved to a different world with a different environment. So it appears that the genetic predisposition also plays a major and a significant role. So interesting thing is that if you would compare that there is a variation across the population. To talk about the easiest would be here to cite an example of certain genes which are known to be significantly altered or associated with lung cancer. Two of these I would just kind of like name. I mean, there are several of those. EGFR, that is an epidermal growth factor receptor. It's a tyrosine kinase, it's an enzyme. And then there is a KRAS. KRAS is a small GTPase protein. So both of these are very significantly and very frequently mutated in lung cancer. But it's very interesting is that their mutation frequency is very population specific. EGFR, to name, is mutated among Caucasian population is 10 to 15%. In the East Asian population, when you would see, the mutation rate is significantly higher. It's 35% and higher. It may go up to even 50% among lung adenocarcinoma patients. When it comes down to India, and even the China falls into lightning, there are several reports which kind of talks about it. There the mutation frequency appears to be, China is more at a higher side. In India, it's more of an intermediate frequency. It is from about 20 to 25%. Which is intermediate to what you get to see in the Caucasian population as compared to the East Asian population, which is a strong indicator that there are genetic determinants which are defined more by the population, by the ethnicity of these folks. And interestingly, the KRAS follows a completely opposite trend. The KRAS, what happens is that the incidence of KRAS frequency is higher in the Caucasian population, and it is reversed when it comes down to the Eastern population. So it's a mixed bag of all of these determinants. So after completing my postdoctoral studies from the Broad Institute of Harvard and MIT in the lab of Dr. Matthew Meyerson, I started my group at ACTRAC, which is the Tata Memorial Center, one of the largest tertiary cancer care center. We defined the baseline mutation frequency to which I just alluded to, comprehensive baseline frequency of the occurrence of EGFR, KRAS, and other different mutations that occurs in that region. We went on to characterize the complete whole. We used the cutting-edge technologies, more contemporary technologies such as next generation sequencing platform. We use these platforms to understand how the genomic alterations that underlies in the Indian lung cancer genome in particular are different from Caucasian, if so any. What we find is something interesting is that the major player remains to be the same. If it is the STK11, if it is the EGFR, if it is the KRAS, they all remain the same, albeit they are occurring at different frequencies. But then that's not the complete story. Is there something which is private and something specifically, or something which is more specific found in the Indian population? Did we find any surprise? Yes, we did. We did find a significant alteration occurring at a frequency of 6.6% in lung adenocarcinoma in another tyrosine kinase gene, that is an FGFR3. So FGFR3 is already known to be mutated in lung cancer, more so in a different subtype, that is a lung squamous, than lung adenos. Lung squamous is more associated with tobacco smoking, lung adenos are relatively less associated with tobacco smoking. What we found that there was a higher incidence or frequency of this novel alteration FGFR3, and interestingly the Korean group, a group from South Korea, also could find the same mutations in lung adenocarcinomas. And this doesn't appear to be there in the Caucasian population. So doing these investigations, that does allow you to explore and understand and contribute to the field more than what kind of like is already is known. And that means is that you cannot just simply adopt or adapt the treatment options or the characterization of the genome, what has been done in the West. In particular, the next generation sequencing technology, to name it, has absolutely been disruptive. I mean, they have completely allowed us to extend the boundaries beyond, not just to understand the biology that underlies these cancer, but also to make it more clinically relevant. And that's where the real strength kind of like comes in. Most of these oncologists in India and across the globe would today agree that these technologies have definitely had a significant impact in their clinical practice. Today, I would imagine that majority of the oncologists, specifically for those who are treating colorectal cancer or they are treating lung cancer in particular, and a few more, NGS or these technologies have become an important part of their arsenal. In fact, with more and more options and more innovative ideas coming in through, such as several of those kind of brought by even the MGI has really making it more and more affordable. It has really kind of given most of the biologists and clinicians a spin that they adopt it or stay behind. Sometimes back people were wanting to use their technologies because it was cool to use. It looked very fancy. So if you would have some additional funds. Today it's more of like their basic line of research and basic line of intervention that is being offered into the clinics. That's the adoption of these technologies have. So I believe that is phenomenal that these technologies are impacted both biological sciences as well as into the clinics.